as I said, I'm now at the Leibniz Institute for Polymer Research in Dresden. This was also where this project was happening and where I was working on this project. The project is called Microplastics. It had a lot of foci around microplastic pollution in the marine environment mostly. And this is a part of this whole project that I'm presenting now, which was, well, you will see in the presentation. Yeah, so let's just jump in. Here, just a quick overview who we are. So this is our analytical chemical research group that is mostly specialized in Raman and FTIR spectroscopy, and focusing very much, but not only on microplastic quantification. And also this project that I already mentioned was a wider consortium, and in there there was one task to build up this spectral database that I will talk about now. So what we do in our research group is we mostly do microplastic quantification. So that means detection and characterization in a quantitative manner of microplastic particles in samples. That could be from the environment, that could be from living tissues, or it could be from food. We have a lot of food samples recently. And we mostly focus on the small part of microplastics. I will go into some definitions afterwards. So that means the particles are not possible to work on manually or even with manual techniques on microscopes, but we need automated methods for which our group developed a software called Gepard. It's open source. You can check it out following this link, or you can try it with a simulation, and you can even use it on your own instrument to steer the microscope and identify particles. And the project, the Microplastics project, had the aim of contributing to the understanding of microplastic weathering, degradation, and fragmentation. So there was a case study of different environmental compartments and sites where microplastics were exposed to environmental conditions. So now to give some guidance and definitions, so we are all on the same page. When we speak about microplastics, we mean synthetic polymeric particles. They enter the environment for various reasons, mostly because of our trash being discarded improperly or being lost and then being fragmented. And we speak about the size range of below 1 millimeter. Some studies also use below 5 millimeter for historic reasons. And we could say down to a lower size of 1 micrometer, below which we would speak about nanoplastics, which again are a bit a topic in itself, because they would require quite different methodologies and different requirements for work processes to work with such small particles. Microplastics are basically an umbrella term. That means it's extremely vast diversity of colors, shapes, material compositions that are or could all be considered microplastics, which makes their analysis and quantification difficult. They are of concern because they are potential carriers of toxic substances that are either added during production for various purposes. Actually, I can hide this little box here. So, and then they could also be sorbed from the environment because, if there are, for example, hydrophobic pollutants or toxins in the environment, they would really like to attach to some microplastic when they are in an aqueous surrounding. Microplastics have also been discussed as a marker for the Anthropocene, for example, as they enter the recently formed depositional layers of the Earth. But now let's focus a bit more on the analytical part. So here is an overview of our work routine, just to give you an impression, and so you know how things are happening in our microplastic analysis. This pipeline starts with that we acquire microscopic images in dark field mode in our case usually, but basically just light microscopy image, but we take it in several set stacks, in a set stack, so several layers to cover small and large particles alike. The particles in our case are on silicon wafer filters. That means we have an extremely plain and smooth surface where the particles were filtered onto, and where we can measure them, their material, of course, but also in their different shape, size, properties. So when we have this image is stitched together in all 3 dimensions to get the sharpest representation of all particles, and then a particle recognition algorithm finds what are particles and what is background and places measurement coordinates in all of the particles. With this, the instrument is steered towards each particle, which can easily be several tens of thousands of particles on such a filter. The filters, by the way, are 1 by 1 centimeters in our case. And then we have obtained at least 1 or several spectra of each particle. These particles are then, these spectra that we obtain are then compared against reference libraries of Raman or FTIR spectra, depending on the technique that we use. Mostly we use Raman spectrometry. And then, a spectroscopist decides software-assisted what material particle is. So of course, this cannot be done for every particle. So we have routines to identify which spectra are certain, or where the software thinks it's quite certain. They are easy and fast to check, to confirm that is actually the polymer that the software reported. And then there is a mid-range where we really have to look into the characteristics of the acquired spectrum. Can we assign this polymer type or not? And then there is a rest where basically the spectrum is just noise, because there was, for example, no real particle. It was just an optical artifact, or it was a material that is not a good Raman scatterer. So then this also would mean that it's not a plastic polymer, because they are all good Raman scatterers. It could have various reasons. And one reason actually is there is a spectrum, but the spectrum didn't fit to the ones that we had in the reference library. Although we try to build continuously our reference library, and there are also big commercial libraries available, the special case of microplastics is that they have been usually exposed to something. They have been exposed to the environment, for example, have been drifting around at sea or wherever, being exposed to sunlight. So the polymeric signature that we see in the spectra could be quite different from what you would expect of this material. So when the process of spectral identification is finished, we have the chance in the Gepard software to look into those particles that were identified as some of the interesting materials, and go through the contours again to see, was the contour recognized correctly, or if not, we can, for example, say, this, so do you see my mouse? This should be actually 2 particles, so we can make it into 2, or we can combine, or we can change the shape, just to confirm that these are right. So we do this step manually for all the confirmed plastic particles, which are usually far below 1% of all particles on the filter that we analyze. And then the results can be reported, either exported as spreadsheets for all the properties of the particles, including the material that was identified, or it can be directly uploaded to a connected database. Yes, so as I mentioned, when the polymers would be very degraded, it would be difficult to always match them correctly with the reference spectra. For this purpose, the Microplastics project set up an in situ experiment where at least for 9 common polymer types, sheets were acquired and then exposed to marine environmental conditions. They have been put into plastic containers that were perforated and then set in these cages. The cages were then exposed at these 3 coastal sites and submerged there, sometimes by divers, and they were in these conditions for several days to months. So from 7 days to 6 months, they were exposed there, and after the different time steps, some were always harvested for the analysis. Some were, after the collection, stored in ethanol to preserve the biofilm that has grown on it. So when they came out, okay, this is maybe not the best image quality, but they looked really nasty sometimes. So sometimes there was a lot of growth, sometimes there was almost no growth. We documented this for each plastic polymer sheet photographically, and then we acquired spectra on each of the sheets with Raman and also with FTIR, ATR spectroscopy on multiple locations of these sheets. After this, the biofilms were removed by digestion with H2O2, and then the spectra were acquired again. So we have a big collection of from several hundred samples, several thousand Raman spectra, several thousand ATR spectra, a lot of images. In total, 300 gigabyte of data, and the analysis was done by 2 institutes that did it both with their techniques, with their devices, with their data reporting routines. And this was basically the point when I entered into the project and was tasked to build a common database from this. To be honest, it was not that bad as is shown in this cartoon, but kind of a messy situation because, for example, one institute decided to store all the metadata in very, very long file names where there were also spelling mistakes and so on. And the other institute stored all the metadata in nested file structures. So similar to file names, but not nested files, nested folder structures or directories. So I could not have one common way to harvest the data, but I had to just implement a script for each of the sub data sets to read in the data and organize the metadata. So I could combine it to one common data set. So I wanted to have the spectral data clear and plain, readable for humans and machines alike. So I just decided a 2-column, so XY column data table stored in CSV files with very little descriptive information in the file name that is like, for example, the polymer type would be there and a few other metadata items, but not everything. And then the rest of the file name is the I just show you this. The rest of the file name that I show here would be the hash of the spectroscopic data content of this file. So I also have a connection between the file name and the data content to be sure that if there's any doubt, if the real spectral data is inside the file, I can just hash it again and check it against the file name. And I see, okay, there was no naming mistake or anything. So in this way, I organized a JSON file that collects all the metadata for every spectrum together with a link to this file name. So together these CSV files link to this JSON common description file form kind of a document-based database system that I can query against with any analytical tool I want. So I put this all together in a repository and snapshotted it on Zenodo. But I also wanted to have the chemical data itself available on the Radar for Chem repository. And actually, I think maybe hopefully after the talk, we can talk a bit about this strategy because I'm not quite happy with this because now I have kind of a double availability of the spectral data, at least. So I have a technical solution by now that I want to implement, but at the time when I published this was the situation. I described that the chemical data is also available on the chemical repository, Radar for Chem, but everything is also available in the code repository that holds all the code that I wrote to make this, and holds the spectra as well. There is also a repository for microplastic spectroscopic data. It's probably not known outside the microplastic community, but I contacted the developers of this, and they were happy about this project and said they want to include our spectral collection there. So yeah, this is also good. Additionally, I wrote an app which was mostly like a quick and fun thing to do, because I already wrote a lot of code for myself to be able to understand the data, to organize it and so on, also to plot it, to see how the spectra actually look like. So I put all of this together in a Streamlit app. If you're not familiar with this, I'm happy to explain a little bit more in the end. I also have a slide in the end to have some recommendations for tools and platforms and concepts that I use here. We can have a look there. So with a Streamlit app, I made basically a quick introduction to what the project was about, how the data was acquired. You have an overview of the metadata, what it means. It shows you all the possible metadata values that each spectrum can have. And then in the bottom, there is a filtering function where you can set certain aspects of the metadata. For example, I want to look only at this specific polymer, or I want to see ATR or Raman spectra of this polymer, and then it plots the data, and you can download directly what you have filtered there only to have these few spectra. You can, of course, always also download the whole spectral collection if you want. Yeah, so you can find the app from the repository, but you can also directly follow this link. And here's a quick view into the app, but we can do a live demo quite just now after the talk. So the idea was that with these filters that you see on the left side, I set the conditions that I'm interested in, and it will plot me if there are spectra available. It will plot them here. Here you see a image also, a photograph also. This is not on the online version because this would have required to store all the gigabytes of photographic material there. So this is only in a local version where I have the photos available. Yeah, but with this, for example, I can now make some comparisons. So, but I will go into exactly what we see here in the live demo. Yeah, what we mostly saw in the result of this activity was that there were many sheets where the spectra looked very similar to the original spectra. There were some where the spectra were completely off, so they would not be recognizable, also not by a human spectroscopist to say this was this material. And then there was some in between, and these are the interesting ones because they can be used then to enrich spectroscopic libraries that are used for matching microplastic potential particles to reference materials. So here we see, for example, a comparison of so LDPE, as it says in the slide title, from the summer campaign after exposure of 90 days with and without biofilm. So as I said before, the biofilm that has grown on there, it has an effect on the spectra that we obtain with these surface active spectroscopy methods, of course. And so in Raman, we see, okay, this is recognizable as a LDPE spectrum, even with the biofilm, but it has some extraneous bands in the middle, very broad, carbon-like, not really specific. This is not visible in the ones that were acquired afterwards without the biofilm. when the biofilm has been removed. If we look at the same spectra that were obtained from the same sheet in ATR, we see a bit more specific picture, because the bands tell us from their positions that these are amide and NH2 bands at their typical locations, and this just speaks for that there is some protein-like biofouling going on the surface, which is expected. We also saw what has already been found in previous studies, that if there was a lot of degradation going on, the spectrum diminishes to either partly or completely. So this is an older paper, but I posted it here because this effect we could see also for some polymers, that you can come to a point when there is a lot of biofilm growing on there, you cannot recognize the spectrum anymore, as what was in this case PP originally. Yeah. Then I'm already through the talk. This is a recommendation slide. I think we can keep this for the discussion. I quickly try to jump into a live demo now. Where it's here. So, yeah, this is how it looks when you open the app. Shows you who made it. It gives the introduction. There is some pictures and even a short video from how the samples were produced. So I just make this small again. And then it gives an overview of how much data is there, the number of spectra that are included now, and from how many different samples they are. I go into this raw data viewer later. First is interesting, the metadata guide. It gives you a listing of what metadata is connected to each spectrum and what values it can take and what it means. Because some of this is just like abbreviations to not expand it with too much text clutter. But with this guide, you can get an easy overview, and it will give you all the different possibilities that you will find here. It also has a polymer guide, because the polymers here are just listed as numbers, but each number stands for a different polymer, and some are also called in situ in the number, and this is because some were not exposed in this way as I showed in the presentation, but some partners collected also what they found on the spot, and this was also analyzed. So they are marked with a different tag in this case. So then the spectroscopic metadata or acquisition details are also listed here. And what I jumped before was a raw data viewer. It basically folds out some data frames that I can open here. So this shows the data tables that are in the background of this app. So you have a chance to actually look in there if you want to find something specific. Gives you some statistics on it. Yeah, so these filters on the left side, I can change them around. So yeah, let's say I want to use this region. This is for Villefranche in France. I want to use, let's say, not spring, but I could also use several or all, but I want to look at summer. Okay, okay, there is an error. I think this is because there is no data available for this filter that I just said. Still, and this exposure days 7 is quite short. So this was just 7 days after they have been submitted to the marine environment. So I want to look at some longer time, 90 days. It's not the fastest. I haven't optimized anything in the code to be fast. This is maybe something that could be improved here. Yeah. And then when we say, okay, maybe I just leave it with PET for now. The one that I showed in the presentation was LDPE. But now that I have set these filters, every time I change something in these filters, this has changed here, and now I see all the spectra that are available that match these filter criteria. So right now we look at ATR spectra. Let's see if there are also Raman spectra. And there are. These are Raman spectra that are hampered quite a lot with fluorescence here. But you can clearly see the difference. All these down here, they would be no bio, so this means biofilm has been removed. I have a hover over pop-up thing where I can go over a specific spectrum, and it will show me all the metadata of this spectrum. Yeah, I could even switch on a ruler, but I even wrote it here. It's a bit slow. If I want to make peak comparison on a specific point, I want to see, okay, so this is at what intensity, and compare if they align with different spectra. And lastly, I can download the spectra that I have filtered here. If I'm specifically interested in these, I can download them as CSV files. So I just see this is measured in nanometer because I just decided I don't want to change the raw data. I will put it into the analysis, into the app in the way I got the raw data, and some Raman spectra were saved as nanometer. Usually we would use a wave number per centimeter. So I put in a little converter, and so now it's in wave numbers, which is the more typical thing to look at for a spectrum. Yeah, this would be the walk through the app. You're welcome to try it out yourself. I'm happy for any suggestions, improvements. Let's open the discussion, I think, or maybe just quickly back to the slideshow. Sorry. Start from this, just to acknowledge all the contributors. Come on. So. This was the project Microplastics and the Leibniz Institute of Polymer Research under this project funded, as you see here. You have my contact details, and I thank you for the opportunity to present it here once again. Actually, I'm presenting right now from the NFDI for Earth plenary, where I was also invited to speak about this data publication. Which I did yesterday, and now I took the chance here in a quiet room to present here at the Stammtisch. Thank you.